The overall goal of this procedure is to introduce a high concentration of fluorescent or ion-sensitive dye into neurons or any neuronal tract using a simple, low-cost technique. This is accomplished by first producing two types of pipettes, type 1 and type 2. Then connect a flexible elastomer tubing to the type 1 pipette and apply positive pressure to draw up the target neuronal tract into the type 1 pipette, verifying that a good seal is made. Next, insert the type 2 pipette into the type 1 pipette and use the type 2 pipette to aspirate the residual ACSF from the type 1 pipette. Then, introduce the dye, but no air bubbles, to the type 1 pipette. The final step is to incubate the neurons in the dark between 6 to 20 hours. Ultimately, after the removal of the type 1 pipette, the preparation is ready for in vitro optophysiological or morphological studies.
The main advantage of this technique over existing methods like bath application, bolus injection, and electroporation is that the dye is delivered to a defined neuronal population. This method can help to answer key questions in the neuroscience field, such as activity, connectivity, morphology, and distribution of defined neurons. Generally, the technique requires practice, particularly ensuring that the dye does not get diluted within the type 1 pipette and no air bubbles are introduced into it.
Begin this procedure by pulling the polyethylene tubing over the flame of an alcohol lamp to produce two short pipettes with tapered tips. First, pull a short type